I wanted to welcome everyone this afternoon. My name is Steve Hoffman. I'm the Director of Inflammation and Immunity in the Research Partnerships Division of the Foundation for the NIH, and really appreciate everyone joining today as we take a bit deeper dive and an overview and the scope of the development of our Accelerated Medicines Partnership Gene Therapy Program as we go forward. Can I have the next slide, please? So today, as I said, we're going to provide an overview of this program, not only about the design and the development of it and many of the aspects that we've been working on with a very broad group of experts and leaders in the field, but at the same time, we also want to provide some greater depth and insight into what the partnering opportunities may well be as this moves forward. I wanted to highlight that we are definitely recording this for archival purposes, and the slides that are presented today we will definitely share back out with all the attendees. Next slide, please. So just some of the important housekeeping. We're using Zoom, obviously, and those that most of us, I think, at now 5 months into all this, are pretty comfortable with the controls of these, but please make sure to mute yourselves on background noise. You may also want to reduce some bandwidth on your videos if you so choose to do that. An important thing is that we're going to collect the questions and comments that you guys may have through the chat feature. So please utilize that as we go through and send those. And during the question and answer sessions at the end of the larger portions of the program, we will read those out. And if we need to unmute a couple folks to have that conversation, we will, but we'll try to capture that as best possible. If we don't get to some of those questions, we're still going to capture all those, and we'll feed back through the leaders here on the group and get back to you with answers and further clarity and follow-up. If there's any problems that occur, there's an email there for our events at fnih.org that our team that is helping us here at FNIH run this. Please feel free to reach out with them if you have any issues with connectivity. Next slide, please. I'm happy and very pleased that the leadership of this is able to present today. Certainly the program director from the Office of Rare Disease Research, Dr. PJ Brooks, has been essential in doing this, and also a lot of the ideation and push from the FDA and CBER from Dr. Peter Mark. So both of them will carry the majority of the presentation. Also, you'll hear from Dr. Joe Manetsky, our Associate Vice President of Research Partnerships at FNIH, and myself on some of the other higher level elements of the partnering, as well as a little bit of where the formation of this project comes from within FNIH and within our Accelerated Medicines Partnership. Next slide, please. So here's really our agenda. Again, that overview we'll provide from FNIH and then get into the nuts and bolts of the program with Peter and PJ carrying that through. We'll then have a time period for some questions and answers that we'll dig in on where your inquiries may be. And then we'll move into a final session around some of the communication efforts that we think will be important here in the partnering opportunities and clarity there. And then a little bit more time for some Q&A, and then wrap up at the end. So really appreciate everyone joining. We hope we'll find this very engaging and helpful. And with that, I want to move and pass the torch to my colleague, Joe Manetsky, to take us through some of the background.
Okay, I'm unmuted and hopefully you can all hear me. Yes, I know you are all here to hear about where we stand with the AMP and gene therapy, and we will get to that. I will go through this very quickly, but I think it's important to understand the context of how this project is being set up and how it will be run. And so that requires me to give you a little bit of background on FNIH. I won't do a lot, but I think it's important that you understand where we are for those of you who don't know what FNIH is, and then with a little bit more detail, what is an Accelerating Medicines Partnership, AMP, which we call AMP. And this is something that how we run the projects is driven through this approach. So with the next slide, we can talk a little bit about FNIH. FNIH was initiated by an act of Congress and has been actively involved in supporting the mission of NIH since approximately 1996. It's actually kind of closer to 25 years really that we've been active. And the way we do this is through setting up large public-private partnerships and working across boundaries that NIH sometimes can't do. And so we hold a very special spot within the research programs that are ongoing within NIH and outside of NIH because of our special mandate from Congress. So there are things that we can do that really no one else can do. And we've done this over the last many years. And NIH, I think, has also found that this is an extremely successful approach, is to do these kinds of big projects that really drive the science and make the results public. So the next slide shows kind of the numbers. It's kind of by the number, FNIH by the numbers slide. We've raised a fair amount of money, nothing compared to the NIH budget, but the money that we raise tends to be very directed towards specific projects. And in fact, the money that we raise actually goes to those projects, and with about 90% of every dollar going to the project and only 10% going into the maintaining of FNIH. Over the many years that we've been active, we've run over 600 projects since the inception. We currently are running 124 that are active right now, which is kind of mind-boggling for someone who's actually even in the foundation, because we're only 70 people. So it's 70 people running that kind of thing is pretty impressive, I think. And even more impressive is that over the last 17 years, we've exceeded industry standards with Charity Navigator, since the foundation is a not-for-profit 501c3. So it's a number, that 17 years is a number we like to hold tight and proud for. The next slide shows the kinds of partnerships that we've done. And I'm not going to read through these, but you can see that we cover everything from global health to specific initiatives within NIH. We run the Biomarkers Consortium, which is a large consortia of consortia, so to speak. LungMap and its predecessor, iSpy2, which was really kind of the driver of the initial thoughts that we do now with master protocols. We've had a hand in HEAL, and sounds kind of good. And we've been recently spending a lot of time on GeneConvene, which is a global collaborative of that many of you may not know about. And then the other thing that obviously has been very active recently, has been consuming most of my time actually, is the accelerating COVID-19 therapeutic interventions and vaccines. The point of this slide is to say that we do and have done many large, complicated partnerships and with extremely successful outcomes. And AMP has been one of these. It's at the top of the list there, and it is been and continues to be a very successful and a productive partnership. The next slide, move on, is just the AMP. This is kind of the transition slide to AMP. So now I've told you a little bit about FNIH, and now we're going to go into AMP. The next slide is an overview of AMP. This was started about 7 years ago, I think, with predominantly by conversations between Dr. Collins and Dr. Dolston, Dr. Collins of NIH, obviously, and Dr. Dolston of Pfizer, and in recognition that there was really a need to increase the number of new diagnostics and new therapies. So there should be an end there. That everyone was spending far too much time and too much cost in developing them. So the approach was, if we got a bunch of people in the room, all the pharmaceutical companies, 10 in this case, and multiple not-for-profits, and the FDA and FNIH in the room that we could generate programs that could actually accomplish these things. And we did that. And 6 years ago, we started 3 new projects: the Alzheimer's disease, type 2 diabetes, and rheumatoid arthritis and lupus. And then, because of those, and I'm not going to go into the results of those projects because they're numerous, and we could spend 2 hours just talking about any one of those. So, but what I'll say is that they have been consistently lauded as a good investment from the companies, as well as incredibly helpful to the field, particularly in each of these spaces. And because of that, we've added on to them with Parkinson's disease, which started a couple of years ago, schizophrenia, which will start this year, and it continues. So, as I said, the first 3 were for 5 years. We're at that end of that. Those are now looking into continuing programs. We have additional new programs, gene therapy being one of them, as well as some initial discussions around heart failure. The important part to understand with this is that each one of these and all of this is being project managed to the hilt by FNIH. We spend a lot of time making sure that milestones are met and go no goes are made. So that's the kind of things that we have done. That's what AMP tries to do. The next slide just gives you an idea of who's been the players for the first 4. And you can see that there's a lot of players. So not just one institute at NIH, but multiple institutes in NIH, FDA's involved, not-for-profits, and a number of different for-profit entities. And NIH, and FNIH manages these large number of partners, and manages the partners in the projects to generate actionable results that are shared with the public almost as soon as they come out. So the embargo type time frame is in many cases 0, and if not 0, it's very short. So this is what we do. I mean, these are the kinds. So what I'm trying to tell you is the thing that we're going to talk about today, the AMP for gene therapy, is not something that is a completely new entity that we haven't thought about how to do this before. We know how to do it. It's actually what we do best, I think. So this is what we do. The next slide is how we do it. It's the operating principles of AMP. We maintain a simple governance with steering committees and under an executive committee. Steering committees nominate co-chairs, etc. There's equal influence from industry and NIH on the steering committee co-chairs. So we have one industry, one NIH. We have extremely firm rules on funding equity and in-kind contributions, and that's all managed by us. There's tight management of deliverables and timelines. So this is very much like a business in some ways, with deliverables and no-goes, and that are defined by interim findings. We expect industry engagement. So it is planned that if you are a partner in these projects, you are an active, vocal partner that will help ensure the focus on the relevant translatable research. It's not just like, here's some money and tell me what you're doing. It's really very much a partnership, as we say. And so we have the patient advocacy groups. We work out data sharing. We make sure all it is pre-competitive. And one of the way we work this out and way we make this happen is that we work out all these important details before the project starts, so that as we move through the project, we have a clear foundation on how to execute. And this is important background because this is exactly what we're here asking of you today, and actually to the beginning of what we're doing today. So the next slide is a bit of the timeline on gene therapy. So this started as an idea at HEVR and with Dr. Collins and the HEVR group, which is a group of senior pharma leadership. I think also included FDA. Dr. Marks was very much involved in that too, as well as Dr. Woodcock. Over the year and a half that we've been working on this, it's gone through and with discussions with a number of different entities like IMI and FPI and others. And we've had a face-to-face meeting about this, as many of you were involved in, last December 9th. At that meeting, we recognized that there were some needs that needed to be generated. We put together working groups, and the working groups really put together a concept that was approved just a couple of months ago, a month and a half ago. And so now where we are today is at this point where we actually start to begin to take that concept to a plan. And so the working groups have been busy, very busy, as they will tell you. We were on calls with them multiple times a week, but unfortunately they're not done. And so the next slide shows where we stand and what we're thinking about here. So we've engaged a number of people, multiple folks from the federal government, NCATS, so these are institutes within NIH as well as FDA and CBER. We have asked for, in this development phase, because that took time and effort, got some commitment of funds to support the development phase for AMP-GT. We've had, besides the commitment of funds, we've also had a tremendous amount of involvement from the partners already, both these industry folks, but a very large number of academic folks that are on the call today, and we'll highlight their names later. But there's been a lot of work. The working groups have, with inspiration, which is I think what I think it meant I meant by the lightning bolt there. We've come up with a concept that's there, but if you'd go to push to the next, this is just the beginning. Next slide, yeah, there we go. This is just the beginning, and now we have to press that go button and build something that's more than just a concept, but actually has defined go, no-go decisions, has very clear budget expectations and timelines. So that's where we are starting right now. The next slide shows you in a slightly different, but maybe more detailed view of how we got here and how actually all AMP programs get here. They start with some sort of idea that comes from the executive committee. We spend a fair amount of time putting together a white paper or a concept. We get preliminary support, and that being at this point, where it's shown here, where it says preliminary support of commitments, it's mostly intellectual, but also in this case it has been somewhat monetary. We are currently at the place now where we have to define a detailed research plan. And that gets done, difference between the preliminary support and the concept and the detailed research plan is that at the detailed research plan stage, we work with NIH, FDA, and our private sector partners. And we do this, and actually, even though a lot of academic KOLs and others have been involved in the setup of the concept, we ask them to excuse themselves from the development of the actual detailed research plan, because during the process of the concept formation, we've invited and have connected with the experts in the field. These are the people who we want to be able to execute on some of this thing. But if they are involved in defining the plan, they would be excluded from potential grantees as part of the plan. So we want them to be our grantees. We want them to be part of the execution of the project. So at this point, as we move forward with the details, we will excuse those. But as we're doing that as well, we will start to talk about and try to get our development folks involved, FNIH, is make sure that we can support the plan as it moves forward, both financially as well as with in-kind and other issues, and make sure that we have connections with the appropriate connections with NIH. So it's all done so that those that could be support the effort are the ones who have the most involvement. So in the past, this has been very much driven by a single disease. And that is, in some ways makes it a little bit easier to follow this path. In the next slide, what you see as we go into the actual discussion of gene therapy is the gene therapy AMP is a little bit of a variation on a theme. It's not a single disease, but it is a single approach to a number of diseases. And so with that, while it's different, it's the same, and it does fit under the AMP banner, and so that's why we're talking about it here. And so with that, I'm going to let Dr. Marks discuss, the initiation and his view of where gene therapy sits right now, and then we can walk through what that variation looks like. So with that, I will move to Dr. Marks. Peter, if you could take over.
Thanks very much. I hope you can hear me now. And let's see if I can take over here, or you can advance the slides for me here. Thanks. So what I just want to kind of give you an idea is why are we so interested in this at FDA? It's because we see the whole scope of gene therapy as it's advancing from big to very big, and down on the other end of things to very small. And at this point, we are noticing that even here you see the increase in gene therapy applications in the top right here, where they almost doubled in a year. You have doubling of the number of applications. And even looking at what's come in over the course of the COVID period, we're still having applications come in at a reasonably good clip. So this is clearly an area where there's a lot of activity. Many of these gene therapies are addressing very small populations, but potentially very effectively addressing the needs in those small populations. So they're products we want to see move forward if we can. And if you look at the bottom left, what you're seeing is what MIT, their NewDigs group, predicts as the commercial success of gene therapy. These are mostly for larger indications with estimating 40 to 60 product launches and 500,000 people treated by 2030. What this doesn't take into account are the literally hundreds of diseases that could potentially benefit from gene therapy, but for whom our current gene therapies are not commercially viable. And if I get the next slide, why is that? Let's look at what we've got here. We have the approved gene therapies right now are Tissuegen Loclucel, Axicabtagene, Veretagene, Onasemnogene. Onasemnogene is Zolgensma. That's the first of the systemically administered gene therapies in the United States. There's actually there's a fifth CAR T cell, which is now approved just recently for mantle cell lymphoma. That's so we have 5 now. In the European Union, you can see they have a few additional autologous products, but notably, they had a product for lipoprotein lipase deficiency, Alipogene, which was approved in 2012, but then withdrawn in 2017, not because of any safety or efficacy concerns, but because it just wasn't commercially viable. So the manufacturer voluntarily withdrew it. So this is what can happen. I go to the next slide, please. So where's the problem here? The problem is that when you're making gene therapies for relatively small numbers of people, you have a situation where there are a number of different challenges. There are challenges in manufacturing, in non-clinical development, in clinical development, and in product access, all because what you're dealing with here are, you might be using in the United States 10, 20, 30 doses a year, which is not commercially viable, really. The commercial viability comes in the sweet spot of maybe anywhere from 100 to a few thousand doses a year. I show you the very large size. So the grande size is our sweet spot. The venti or large size is still something for the future, because gene therapies that would be systemically given to adults made on the scale of tens of thousands of doses are still a while away. We still don't, on our current technologies, if we were to go in that direction, we'd probably need bioreactors the size of Lake Erie to take care of this. So they're a little bit away until we get to more efficient methods of producing them. I know that's a little hyperbole, but you get my drift. On the other hand, though, there's this, what we've really come to be interested in at FDA are these gene therapies that could potentially be transformative for a group of patients who currently has either no or very few alternatives. And that is these gene therapies that are individualized or bespoke gene therapies for a few or one or a few people. And here, the problem is really one, not that we can't make them, it's just we can't make them efficiently enough so that they can be commercially viable. So we need to find a way forward now so that we can see patients who are potentially treatable with something we can do now, be able to get those therapies. And that's the issue that we're trying to get at, the heart of the issue. And so I think, I'll take one, this next slide, please. So the issue here, before I turn this over, I just, really the idea here is to try to make this technology of adeno-associated virus as a vector more accessible to a broader range of diseases, because it really does potentially allow itself to be used for a variety of different rare genetic diseases, or not so rare diseases as well. We think it could potentially streamline, what we could do in a partnership is potentially streamline the non-clinical and the product testing that's needed. The idea being that if one were to use the same vector, treat the vector almost like a razor, the handle to a razor, and start treating the inserts like the razor blade, that you would not you wouldn't recheck the razor each time. You would just have to understand the razor blade. And then we also think that we could hopefully, with this partnership, start to facilitate scientific advances in AAV biology that could ultimately benefit the entire field, as well as in the manufacturing technologies that really the idea here would be to see if we could bring them down in cost by almost an order of magnitude, which would then make these therapies actually very cost effective. And most importantly, I think we'd like to see therapies brought to the individuals who need them sooner. So with that, PJ Brooks, who's the director of the Rare Diseases Research at the NCATS, or the National Center for Advancing Translational Sciences, I think he's going to pick up, and then we'll tag team it, and I'll come back in a little bit.
Great, thanks, Peter. If I could have the next slide. So this is an overview of the whole AMP gene therapy. As you can see, there's basically two major components to it. On the top is AAV basic biology, but particularly basic biology focused on AAV gene therapy. It's not basic biology just for the sake of basic biology. And down below is advancing access to AAV technologies and vectors for bespoke clinical applications, what we call the Bespoke Gene Therapy Consortium, or BGTC. On the basic biology, there's two components to that actually. One is focused on enhancing vector generation, and the second is on enhancing therapeutic gene expression in patients, which is ultimately the therapeutic benefit. And in the Bespoke Gene Therapy Consortium, and there's a certain parallelism here because there's a component focused on AAV vector manufacturing and analytics, and also a component of the BGTC focused on clinical development. And as you can see, we anticipate that these two different components will have a lot of crosstalk and ultimately influence each other. Next slide. So getting more into the basic biology, as Joe mentioned, we have various working groups, two of them, one focused on vector generation and one on gene expression. And I'm not going to read through all the names, but just want to highlight the fact that we had a lot of people from different parts of the NIH and also industry involved in these different working groups, which are very much focused on identifying questions that could be asked that are pre-competitive, which is an essential part of this concept. Next slide. So for the advancing AAV biology, one question is around enhancing vector generation. As you can see on the right, the way we make recombinant AAV vectors for gene therapy is we start with plasmids or viruses or other things that contain the helper function as well as a therapeutic gene construct. Those are transfected into producer cells and out comes recombinant AAV vectors. But in fact, within there's a variety of specific mechanistic steps, some of which are shown on the left. And the important point is that each one of these steps have not been optimized to be the most effective in making recombinant AAV vectors. But they are specific mechanism steps that could be addressed individually. Next slide. And again, when you think about what happens when a patient gets AAV gene therapy, the vector infects the cell, but ultimately there are many steps involved between the infection of that cell and expression of the therapeutic gene, which is what you want. That's the therapy. And here again, there are individual specific steps that can be identified and could potentially be optimized, but as yet have not been optimized. Next slide. So the approach that the working group came to is to support high throughput functional genomic approaches as well as some targeted funding opportunities. And specifically, we're interested in having assays for each step in the AAV vector production, gene expression, target cells, those individual steps I mentioned before, but to have assays developed for each of those that would be amenable to high throughput screening. And then this screening could be carried out either whole genome screening with siRNA or CRISPR approaches, or a library of approved drugs available in the public domain. And as we think about doing this, we anticipate that at least some of this work may be done at the NCATS intramural testing facility, which has a tremendous capacity for carrying out these high throughput screening assays using advanced robotics technologies. But also that's not going to be the whole thing. We have left open the possibility that we may also have some focused funding opportunities on specific questions that are just not well characterized or covered by the high throughput screening approaches. Next slide. And so in terms of the key role of the partners, the gene therapy partners in the AAV biology component, they will have the opportunity to prioritize and weight decisions and weight questions in primary focus, to shape the format and process for sharing the results, providing continuous guidance of the research through periodic review of the projects, and of course suggest innovations to the BGTC pilot program. So I'm going to hand it back to Peter now to talk about the BGTC.
Okay, thanks very much. So the idea here of the Bespoke gene therapy pilot program is that we'd very much like to see how we can do all of these things that we were talking about in a way that will help us get access to these AAV technologies for these specialized bespoke clinical applications, again, from a few people to it could be larger populations. I think as we all know from working with rare diseases, one of the things we should understand here is that what is very rare today may be less rare tomorrow, because once one has a therapy for something, oftentimes patients start to come out of the woodwork. The idea here is to join what we'll do on the side of scientific work and clinical development, and then this Bespoke Gene Therapy Consortium to actually get advances to patients sooner. If I could have the next slide. So under this kind of umbrella of a Bespoke Gene Therapy Consortium, or BGTC, the idea here is under one roof to go from the concept of I have a gene that I can target for a rare disease, make that vector that targets that disease and the correction of that disease or the treatment of that disease, be able to manufacture it, and then being able to get it to patients to treat them clinically, gather data, and ultimately to get it to them sustainably. That's what is in the center of the slide, from the head on the left to the vaccine looking thing on the right. So the syringe on the right. So the idea here is that how would you get there? Well, the way to get there from our perspective at FDA is to start to standardize things, make use of the fact that you're using that razor and you're inserting different razor blades. So you don't have to, once you know enough about the razor, you don't have to reinvent it every time. And the idea here then is one for instance, with vectors, you'll understand a standard menu of vectors that you can use. That standard menu will be well characterized. You'll have data such that you can have master files with FDA so that one doesn't have to redo all the preclinical, non-clinical work and the vector characterization. One simply uses it and then has to just understand the insert or the razor blade that you're using. We'd also like to try to use existing manufacturing technologies and purification procedures, so that doesn't have to be redescribed. One can just reference a previous file. And then in terms of the clinical work, many of these, although there will certainly be unique outcome measures, a lot of this work could be done in a way that is able to be reused and repurposed. So the idea here is to try to do as much as we can from start to finish, and do that. The thought here is under a nonprofit umbrella organization, which for at least these trial runs would allow the facilities for vector generation to exist, small batch manufacture, and help start to do the improvement of manufacturing efficiency, and ultimately potentially out-license things that could be commercially viable. One of the things that I think that is sometimes lost on this is one of the things we really have to gain here is by using a standardized process and putting a lot of things through it, we are going to start to improve in a way that we simply don't right now. Because when you do things as onesies and twosies, you never continuously improve as you go along. And I think if we put multiple things through the same process, we'll understand not just how to improve the manufacturing, we'll also understand how to improve the throughput through the system and how we actually do some of the regulatory things along with this, and we can hone that and make that more streamlined, ultimately, hopefully allowing things to get to people more quickly. Can I have the next slide, please? So this proposed consortium, the idea here is just really ultimately almost have a playbook, a simple manual for doing this. My thought, because I trained in the kind of the time of molecular biology when everyone had their own copy of Maniatis, but this would kind of be the Maniatis for gene therapy in humans as opposed to cloning in bacteria. But this is, I think, something if we did, we could understand then by that playbook would be put together and would allow us to run pilot projects for some different diseases with different prevalence, vector dose requirements, different routes of administration. That would help us to get some understanding here of the constraints on production. The idea here is not to use an incredible array of vectors, but to really concentrate on a couple of different serotypes so to be able to reuse and leverage the different vectors. To try to make sure we identify the appropriate target diseases very carefully through a process that the clinicians use and in the analytic approach. And as we're doing this, define and standardize the vector quantification, the toxicity testing, lot release assays, and do that in a way that we have industry and academic manufacturers collaborating, so we kind of start to come to a harmonized approach. Can I have the next slide, please? So these are just to show you the, I'm not going to go through every name here, but just to show you the BGTC working groups, one on manufacturing and analytics, the other on clinical development. They have done a tremendous amount of thinking and work on this and have really, I think, helped refine this concept and validated that there does seem to be a very real need here, and there really is this opportunity to make some significant progress rather quickly if we have people join together. If I could have the next slide, please. Okay. So, you know, what's the strategy for leveraging expertise and capacity to manufacture these gene therapies for the pilots? The idea here was to try to have a couple made by industrial manufacturers, a couple made by academic manufacturers, so we could see how both processes worked. The industry brings the experience and expertise, and in some cases, the history of approved products. The idea is then you take a couple of serotypes, move them through the process, move them through to different target tissues. And the hope here is to try, for instance, to isolate this so that the transgene sequence is the only difference being introduced into an established manufacturing paradigm. The idea is that way, what we can say is we can leverage what is known about the vector and the manufacturing of that vector as much as we can. Now, obviously, there'll have to be some guardrails on the size of the insert, because we know if you can put something too small or too large, you can affect packaging. But to a first approximation, we think this is a way to think about this. And then the next slide, please. In terms of the vector manufacturing, we like to prepare and test vector supplies for both the preclinical tox studies that need to be done, as well as clinical use. Again, how much of that's going to need to be done in terms of the preclinical work will depend on the exact application and what's been done previously. And the idea here then is that with one batch, one would like to have enough of this vector to use in a clinical trial. In some cases, that might be 20 doses, and some it might be 40 doses, but you'd have a batch that would get you through this, this development. And we can talk more about the concept for using Bayesian clinical trials to get that done, but you can see that depending on where the gene therapy is being given, we might need, you know, different scales of manufacturing here, ranging from 10 to the 13th to 10 to the 15th viral genomes per dose. The idea is for partners here to be able to pick something that works for them, that they're doing already, and then the manufacturer supplies that this product, and we're really looking right now at the willingness of people to supply this in kind support for the pilot projects. And I think actually, hopefully, we will see this come to fruition, because I think there seems to be some enthusiasm here. Go to the next slide for me, please. Release testing and specification, obviously those are very important, and we increasingly think that the more we can identify in terms of critical quality attributes and go through them, that will be very important. Again, the idea here is to try to use this as an opportunity to reduce the burden on what needs to be done, understand what's really important and has to happen, and what can we do without, so that we can actually get better throughput at lower cost to be able to help more patients. the idea here also is to describe, what tests would be needed. This is all kind of going into this Maniatis playbook or cookbook, so that we can do things the same way over and over again. And I'm just going to take a moment to digress here. The molecular biology analogy for Maniatis is not lost on me, because after about the 30th time I made bacterial lysis solution according to Maniatis, I figured out, oh, well, I adjust the pH just a little differently and I get a much bigger yield. Okay, great. I know that. The reason why I was able to know that is because I did it 30 times. Now, I'm not saying we're going to have to do this 30 times, but what I'm trying to say is that by doing the same thing according to a procedure that you follow in a standardized manner, we can hopefully continually make improvements, and ultimately those improvements, I think, will be exportable to help the larger field. And I think the field could actually be larger than we think it is, because when you start to think about vectors that will carry genome editors, it's going to get larger. And whether that's using an AAV vector or some other vector in the future, we need to have the paradigm ready. If I could have the next slide. So obviously, I'm not going to spend a lot of time on this. Standardize, standardize, standardize. The goal here for assays is to standardize them. Our perspective right now for the vectors that people contribute, manufacturers will test the vectors using their standardized processes, and there may need to be additional support to fulfill these requirements. But there'll be some centralized testing facility that NIH would establish to conduct supplemental testing and product-specific testing, such as potency. So as the IND holder, NIH would be responsible for the certificate of analysis that would be submitted to FDA to support the clinical trial. The idea here is not to overburden those who are generously supporting this. And the final slide that I'll take before turning it back over to PJ is whoops, next slide, sorry, 30. So it's just on engagement with FDA, which is that from our perspective, what can we do? Well, we want to get out of the way of the science and mainly try to help by starting to think about what we can do by establishing master files, having early engagement to deal with specific issues that might come up. And we really, our goal as being part of this project is to try to focus on general principles so we can provide people with generalizable feedback. That's not to say that we won't provide product-specific feedback for individual products, but we'd like to try to develop more generalizable feedback that we can provide manufacturers, and thus help not just those that are participating in the pilot, but others as well. And we want to try ultimately to streamline our regulatory requirements so that more people will take gene therapies through this pathway and concentrate on this. And part of that will be by allowing and by facilitating kind of an innovative clinical trial design program for bespoke products. The concept here that we've been thinking about, and it's in development, is one where one thinks about, instead of thinking about phase 1, 2, 3, simply think about rare disease development on a continuum from no evidence that this product works before you've given it to a single patient to, well, some evidence after you've given it to 5 patients and 4 have responded, and perhaps meeting our efficacy standard once you've given it to 10 patients and 9 have responded. Those are just, by the way, those are just hypothetical numbers. That's not guidance. It's simply hypothetical, to the idea being that the idea being not having to get so hung up about what phase clinical trial you were doing, and more into what are the results and do they support efficacy and safety of the product. And so that is really what we're thinking about in terms of how you get to a pathway to licensure. And with that, I will turn this back over to PJ.
Great, thanks, Peter. So if you go to the next slide. This kind of shows an overview of the whole BGTC, with kind of what Peter described. If you think about on the left, we've got different vector manufacturers, some academic, some industry, making a vector for a particular disease or the certificate of analysis, and that would be then handed over to an entity that we call the BGTC Coordination Center. And this would be coordinated and supported by NCATS and carry out a variety of functions in the whole project. This would oversee the supplemental testing component that would be used to harmonize the data from different vectors, as well as allow the toxicology and other data that would be necessary for an IND submission. There'd be a regulatory core that would actually prepare and carry out and submit the IND to the FDA. I really want to highlight the point that, as you see in this diagram, we put the FDA outside the blue box, which is outside the BGTC, to highlight the point that when these regulatory packages go in, they will go in and be handled exactly like an NIH-sponsored clinical trial, no different than that. So the regulatory process there will not change. Other components of the coordination center will be a clinical trials core that would support a central IRB, interface with the clinical sites, and hopefully carry out these trials in a way as sort of described by Peter, where we can maybe get beyond this phase 1, phase 2, phase 3 sort of process that I think doesn't really fit the way we're doing gene therapy these days. There would also be feedback from the data from the clinical sites with outcomes and SAEs to be matched up with the data from the supplemental testing. So this is kind of just a broad overview of the whole BGTC effort. Next slide. So let me now switch and talk about some thoughts we have on how to prioritize rare diseases in the BGTC. And really important to highlight here that the whole goal of this is this iterative learning process of the BGTC. It's not just to develop 6 gene therapies for the sake of doing it. It's to learn something. And so those are the main considerations on prioritizing and selecting diseases. Next slide. So in principle, there's thousands of monogenic diseases that might be amenable to gene therapy, and the basic idea would be to sort of winnow those down through a process that involves analysis of different aspects of the disease, even things like the size of the gene and the cDNA. Does it fit into an AAV vector? And ultimately get these down to a manageable level for manual review and final selection according to various criteria, including the benefit of the using that disease for the BGTC pilot. And so we had a variety of draft criteria that have been developed through our working group with representatives from government, industry, academics, and bioethics, importantly, as well as patient advocacy. And that criteria is still in progress. In terms of thinking about the BGTC, the partners would have some direct input and role in the final disease selection. And again, highlighting that the goal of this whole project is to learn about the process and improve it as it matures. Next slide. So this sort of shows some of these disease selection criteria we considered. And again, this is sort of work in progress, but some of them are fairly obvious. For example, we want to be looking at diseases that are amenable to AAV gene therapy, typically autosomal recessive or X-linked disorders. Importantly, also diseases that are of no known commercial interest, because that's an important aspect of the whole effort. And also various other considerations there, including being sure we have representation of different disadvantaged populations, racial diversity, socioeconomic diversity, to address those important issues also as we think about disease selection. Next slide. So once you've kind of gotten and winnowed down the number of diseases, there'll be a variety of other considerations in making those final decisions and selections, very much including some ethical considerations. And we've already engaged the NIH bioethics department as we thought through how we would make these decisions and anticipate continuing to have input from them as we go along, in addition to input from the clinical development working group, to be sure that the process of disease selection is transparent and clear, and that is communicated to the community, because there's obvious sensitivity here. And keeping in mind these different core ethical values of research priority setting, and ultimately how the choice of a specific disease impacts the BGTC pilot and our opportunity for learning. Next slide. And just saying a little bit more about the clinical trial design and development and how this should all be done. I think wherever possible, we would like to leverage already existing NIH-funded clinical trial programs, including the Rare Disease Clinical Research Network that our office coordinates along with many other NIH institutes and centers. NeuroNext is another one from NINDS, the CTSA program. I didn't put on here, but of course the NIH Clinical Center is another one, but also other efforts, and certainly not, I don't mean to say we would limit these to those that are funded by NIH. There may be other clinical trial sites that are already existing that are not funded by NIH that happen to be really well suited for a particular disease. But I think the point is that we want to be doing these clinical trials in the context of environment that is well set up for clinical trials. So there'll also be a single IRB to oversee all the projects in the BGTC. There'll be a board of expert clinicians who are expert in AAV gene therapy trials to guide treatment and learning because these would be multi-site trials. We also would like to pilot test the use of visiting research nurses that would allow administration of gene therapy in areas outside of the major academic medical centers to really sort of expand the reach of gene therapy under this program. Next slide. So the key deliverables are listed here. A menu of AAV vector and vector generation processes, harmonized set of vector quantitation and lot release assays, a process for disease selection for trials, this manual of AAV gene therapy and for selected serotype vectors, as Peter mentioned, the Board of Expert Clinicians, importantly, a framework to manage the IND and legal liabilities in these multi-site trials, and pilot studies for shared learning and future processes, and of course, implementing the results from the understanding of AAV biology. Next slide. And then, the key role of the gene therapy partners in this pilot will be to basically participate in this effort and to learn firsthand and guide the pilot program as it moves forward, to engage on sub-teams that are directly involved in the development and standardization and harmonization of testing process, assays, and regulatory interactions. Continuous guidance of the BGT's progress through periodic review of the project, and incorporate innovations and understanding from the AAV biology, which we do think is an important aspect of the whole project, as shown on that last graphic there. So I think that's the last slide I have. And I'm going to hand it now over to Joe, I believe.
Yes. Host has muted me. Can you hear me now?
Yep,
Okay.
can hear you fine.
Very good. So I get to talk about the budget. That's why PJ and Peter dropped off. So the proposed budget shown on this slide, it's very high level because we haven't gotten any of the details. That's our next step. But it's an ambitious project. I mean, this is a very ambitious goal, the outputs that we were just talking about. And it is a very large ask. But it is important to start somewhere. The other thing that's important to really understand is that all of these steps, each part of what we're talking about is actually something that is already being done, but just not at this scale and not coordinated. And so that's really where a lot of this is going. It's not that we can't do this. It's that the putting it all together is going to take a lot of work, continued work. We've split out the budget by the two sections that we've talked about, the host and viral genome biology efforts, which is currently, we're thinking is going to be something like $10 million a year. And for 5 years, that's 50. And then for manufacture, currently with respect to 5 or 6 gene therapies, we're thinking, kind of in that same range, maybe $40 million over the 5 years. And then FNIH is a guesstimate, and we're just trying to figure out what we would do. But it all ends up being about, $100,000. And we will be looking in terms of the partnership, we'll seek co-investment of about 50-50 from public and private sector partners. So, the overall, what we will be asking from our private partners, both industry as well as advocacy groups and others, things like that would be about 50. And then we look for federal support from NIH and FDA. It always comes down to how do we do this, and have we done it before? I told you at the beginning, we've done this before, so we know how we can manage this kind of approach. The next slide shows how, which is we will be managing the decision support and partner engagement very closely. The project plan will expand on the questions that we've talked about, and then actually start generating the beginnings of RFPs that either NIH or FNIH could release and ask for response to. Proposals will be supported and can be supported by multiple funding mechanisms. And the AMP-GT stakeholder representatives will have a role in making decisions on reviewing, prioritizing, and what the best proposals are. We have done the actual dollar funding proposals a number of different ways. FNIH can bring in the money from the private partners and just hand that over to NIH or the FDA and have them do their normal RFP process. The other possibility is that we can take that and supplement the NIH grantees with additional funds in a separate research collaboration agreement, or it doesn't have to be an NIH grantee. We could, provide research collaboration agreements to additional outside grantees or contractors. And in general, our direct in-kind contributions have to be something that takes dollars off of the plan. So something that the plan has been defined, and then there is and the cost of a flask is $100, and you're going to give a flask. You can account that $100 to the in-kind contribution. So that's the very straightforward approach. When we're thinking about this, while we have separated out for costing purposes, budgeting purposes, the two separate or the two general areas within AMP-GT, it's one. There will be one AMP-GT, and the money coming in, the support in kind, whatever it is, coming in will support AMP-GT in total, and that's the current plan. So that addresses some of the thoughts of people who want to get into gene therapy. How could that, but don't have anything to provide in terms of in kind, they certainly can provide funding. So that's I think the final slide before we get to the question and answer period. So I think you've got all that you could probably take for an hour and a half. It's like drinking from a fire hose in most cases. And I think at this point now, I'm going to ask Steve to lead the question and answer period as we move forward. We've gotten lots of questions, and that's great, and we'll try to address them over the next 20 minutes or so.
Thank you, Joe, and also thank you to Peter and PJ for going through what we know is a lot of detail. Some of this we may have commented on, and I'm going to work off. It might bounce a little bit. I'll try to associate the questions as best I can around the same topic. I know there's some crossover on them. But the one comment that came early from Scott May from Lilly was about, can we comment further on the interaction between the AAV biology and the Bespoke Gene Therapy Consortium? How would they, generation of data from the one, which may take some time to then weigh into the BGTC, and vice versa? How do we see that interface interacting? How will we move forward on the clinical development aspect as we know that will begin, and then they interact as the time goes on? I can certainly make comments, but I'll ask Peter and PJ to take that on first.
Yeah, I think Scott's exactly right about that. I do think there would be sort of a delay in terms of that interaction because it will take time to generate the data from the basic biology before it can impact the clinical work. So I would anticipate it would be maybe in year 2 or 3 for just that reason. So that's accurate.
Yeah, that sounds right to me. I mean, I think the nice thing is that from the standpoint of getting moving on the manufacturing piece, the manufacturing clinical piece, regulatory piece, that is ready to go. And I think the nice thing is many of the things that will plow the road for applying the other advances that come along 2 or 3 years later.
And I think one thing that Peter has said and PJ have said repeatedly is that the BGTC will not start with 5 or 6 therapies. It will start with 1 or 2, and then those will get started, and we'll try to learn something from those, and we'll maybe bring in another one in the next year, and then or 2, and then another 1 or 2 in the next year. So there will be time, in the 3rd year or so, or even later, to incorporate learnings from the biology from the first 2 or 3 years.
Thank you, Joe. A couple of the questions address contributions and partnership. One in fact around the manufacturing. If you are an organization that is not yet matured in that internal manufacturing capabilities, how would you still provide expertise and involvement and partnering to the program? And then perhaps similarly, we could touch base on the AAV biology side of this. Is where do we see decisional involvement? Where do we see the partners having representation and engagement on both sides of this if they were a contributor? Peter or PJ first.
Okay, could you repeat the question? I'm sorry, it broke up on me.
My apologies.
Not yours, it was on my end, sorry.
Where can the partners contribute? In one case of if you join the partnership and you don't have matured internal manufacturing capabilities, how could you still lend your expertise and be engaged and involved on the clinical development side of this, as well as then also, I think the same question on the AAV biology, where would you be involved in that triaging and decision making on those questions?
Yeah, thank you. Now that's where I started to hear everything. Sorry, I needed the beginning of the question. I apologize. So let me say this, and then I'll turn it over to PJ. I think there's a lot of opportunity here, even if you don't have manufacturing in place. I think we have enough to do in terms of if you might have a preclinical program in place. I think we need better preclinical, non-clinical models to help us understand how things are moving forward. I think we need people who are willing to think through with us, how will we streamline clinical trials? How can we pave the road with gathering data so that these rare populations, when clinical trials do start, they're able to most efficiently step into them. And I also think we'll probably be listening for what regulatory changes might be needed or adjustments might be needed to accommodate things. PJ, anything?
Yeah, I think that's basically right. I think that if an entity is contributing funds, there's certainly a variety of ways that they contribute to either the clinical development or basic biology, depending on their specific expertise. And certainly have that opportunity would be welcomed.
And I think the only thing that I would add from the FNIH perspective is that certainly there will be, decisions to get specific on, say, the AAV biology, is that there may be multiple mechanisms that we do that. We could do that through a pretty standard government NIH funding perspective, but they also, we didn't go into it in depth, Joe touched on it there at the end, that we also can do funding through agreements through the FNIH, and the triage of those, the prioritization and ranking and level of funding and final decisions of what questions would be answered, what those awards would look like, and the parameters of qualifying those awards would be part of the steering committee, and the representatives would make those decisions. And then similarly, I think on the clinical development side in particular, we touched on a little bit of this algorithm or template to make decisions on disease selection, and when we'll do as much as we can under a process and a well-vetted and ethical outline, but they will get to the point that we'll get to the final decisions on what we would put into the pilot and what we would match that up and through what organizations. And I think that the steering committee and or the representatives of different sub-teams would have the ability to weigh in directly on those final decisions. So we talked a lot about go, no go decisions and decisional control that these representatives will have. That is certainly part and parcel with these types of partnerships and what those roles those groups will have. There are a couple of specific questions that I want to highlight, and then I might need some people to follow up on. There was a question from Dr. Hayashi from Lilly around the high throughput screening, the what target cells are to be used, and is the purpose to study production or viral uptake?
Part of it would be to study production, so these would be in the major human cells that are used for production, also insect cells. So it's like the way you planned it. So I think that, yeah, so I think the hex cells. But I mean a human cell line that is used for producing virus and also an insect cell line. And then for the second component, they would be cell lines that are amenable to transduction by AAV.
Very good. Deborah Miller asked about how the BGTC may work with developers of novel AAV products.
Yeah, I think there's also a second question about how that's similar in that in terms of construct engineering cellular tropism, et cetera. I think
yeah, you want me to? I think the short answer is that this effort is really intended on using vectors that have already been used in humans. We had some consideration about that, but we wanted all of this effort to be in the pre-competitive space. And so that's why we decided not to focus on novel capsids or novel capsids. So that we're thinking about using vectors that are already in clinical use. Peter, you want to add anything to that?
No, I think this is right. I mean, part of the goal here is one of the things that obviously we didn't speak a lot about, but we could start to speak a lot about is the fact that the only thing that rivals the cost of producing gene therapies at contract manufacturers is the cost of attorneys' fees for putting together all the IP issues that could go into this if we're not careful. So we need to be very careful here about what we use for intellectual property, and we want to try to keep things as free and clear as we can. Not to say that we couldn't actually have this, and this is another thing for discussion, this whole process may ultimately generate some very valuable intellectual property or potentially even patentable things, and I think that will have to be addressed. You know, let's see where it all goes. But I think we should probably start. I think to a certain extent, we'd like to walk before we run. I'd love to get to running here, but we just like to. Yeah. I think some of this is walking before we can run.
Thank you. And that also, there's been some follow-up questions about just that. So maybe just not mature enough in our full project plan, but we understand that those questions are before us, and how we'll handle those IP issues is certainly going to be an engaging part of the conversation between the representatives. There are several questions that I think relate to, the use of biomarkers in the selection of disease, natural history, and other parameters, which I know that we could address if we did a deeper dive into some of the questions that we've delved into with the NIH ethics groups, with some of the work that our working group has done there in clinical development. I didn't know if we wanted to, if you could expand a little bit on a couple of those parameters that we had talked about that would answer some of these questions of what are criteria that we're considering and involving for those disease selection.
Yeah, I could take a shot at that. I think we don't anticipate supporting new natural history studies. There are a lot of diseases where there is sufficient natural history data, including many in our rare disease clinical research network, that would be potentially amenable to gene therapy. They're just sort of sitting there waiting for someone to come and do it. So I think we would really want to be focusing on diseases like that, where there's an opportunity and maybe it's not moved forward just because there isn't sufficient commercial interest, which I think gets to both of these things. But in terms of the biomarkers, of course, you'd want to have enough biomarkers or appropriate biomarkers to allow you to do a valid clinical trial and have valid outcome measures. If you had no valid outcome measures, that would sort of rule out that disease. Peter, you want to add to that?
No, I think you got it.
Okay.
But that is some of the learning, right? That as we walk through these and we'll realize what kind of natural history studies do we need and what kind of measures do we need, so that the next group that realizes they need a natural history study does the right one, as opposed to collecting a lot of the wrong data. So again, it's all part of the learning process.
Very good. There are several questions around budget and contributions and how that is broken out.
Sorry.
And no, it's okay. And I want folks to understand. I think we mentioned at times, but it was on different slides. And the budget that was outlined and proposed in the concept form, where we get to the 102 million, really had about 50 million of that going to the AAV biology portion of this, and the remaining funds involved in the overall management and or the Bespoke Gene Therapy Consortium. Obviously, we're at a point of needing to understand where we may have some in-kind contributions on the clinical side from manufacturers, that those numbers could fluctuate a little bit. But really, we see this as one program, and the one program that's benefiting each other, and learnings from the one benefiting the other, and vice versa. So really, the partnering opportunity here is to support the whole program and the benefits as the whole. And the idea is that if you would be a supporting partner, whether it's a pharma partner, or we're going to talk in the next section of the session of the meeting in the webinar about some of the other tiers and levels of partnering that will be available, those full members that have full voting rights and full members of the program will certainly have an equity and access to the information and sharing, but there'll still be many other mechanisms that we think we'll be able to share with the community. And really the goal of FNIH, the goal of AMP, and any of our programs is really to get the information out as quickly and as public as possible. So I think through all the various sources, you'll see us doing that. Joe, I didn't know if you wanted to add more to that, and then see if the others may.
No, that's what I wanted to say. 102 over 5 years, not 500,000, 102. And that is to be refined. So it could be a little more, could be a little less. That's what we need the next step to do. And I think, we really believe, and particularly Dr. Collins believes that this is a single package that has clear interactions between the two that are absolutely necessary in order for the whole package to work. And so it's really a single AMP. It's not individual AMPs, the biology versus BGTC.
Right, and of course, and so everybody's completely clear that the budget is 102 million total over 5 years,
Correct.
not 102 million per year for 5 years.
Correct.
Very good. There's a question that gets a little detailed. Luke, I don't know if we'll need to dig deeper, but maybe see if I can represent it right. Will the assessment of different sources of manufacturing, industry, academia be done per program or only for the first few, and then identified a preferred manufacturer or a process. We want to clarify that. Meaning,
Well, this is Peter. I'll try on this one. That's a really great question. I think we are looking to see, I think we're looking to see how putting through several products through the process works, understanding whether we can settle down on some unified process. Ultimately, I don't think we're going to be able to go back to the same manufacturer over and over again and get in-kind donations over and over. So the idea would ultimately be to settle down on a process that could be done either by a contract manufacturer or an academic contract manufacturer, or a few of them, to settle down on a few manufacturers that could continue to put these through in a way that was essentially controlled and as harmonized from time to time as it could be. I'll turn it over to PJ, but I think that this is the part of the issue that we're trying to sort out at the early part of the pilot, is trying to get there to understand what are the parameters that we need to take into account. What will facilitate the most facile manufacturing at a given site?
Yeah, I don't have a lot to add to that. I think, you know, we're trying to figure out how to get the pilot to work initially, and once we've done that, then we might be in a position to think about how to go forward with the other, you know, beyond that.
very good. And some of that impacts the timeline. There was a question directly about when do we expect the first the first IND, which is obviously a big question mark of launch and getting this together and getting it supported. But somewhat related to that, there was also a question, which I do think we addressed, that NIH would be the IND holder. And you know, some efforts and questions have come from partners either on this webinar, but obviously in individual conversations about indemnification, liability, those types of things. Peter, I didn't know if, and PJ, I didn't know if you wanted to speak a little bit about that on the IND side.
I think only that these are important issues that we're going to have to work out, and I don't have anything more definitive or intelligent to say than that, but obviously we're well aware of them and the importance of them.
Same here.
Good. A couple maybe more specific questions around process. We have a question around, is it possible to look at constructing stable cell lines for AAV production, right? We get into some of the nuts and bolts of questions we may have, or ways to harmonize, streamline. How will we address those types of capabilities?
So this is, yeah, go ahead, PJ. I mean, I think this is, I'm going to turn over to PJ quickly, but this is where this harmony between having these two aspects, I think for practical purpose, the things that go through first might not use that. It may use what we have right now, which are the tools we have, but ultimately you could imagine development into this, and that could be one of the benefits that could come out of this type of project. But go ahead, PJ, sorry.
No, I was just going to say that seems like the kind of thing that we would have probably not been inclined to think about, because it doesn't seem like it's pre-competitive. You could imagine a cell line like that being very valuable, but I guess, you know, it's an interesting idea. I'd leave up to Steve and Joe if there was a sense that creating such a cell line would be a good idea here. I do think there's a lot of people already working on this, and we've seen progress on it, so I'm not sure it's the most relevant thing for this group to do.
Yeah, I think the idea, I mean, would be to understand, use this, the pilot and the biology, to understand as many parameters of what's important for that razor handle. I'm going to hang on to that analogy because I like it, because there may be situations where, you know, in 5 years we find out that you can get the exact same razor handle from a cell line that's been engineered versus a triple transfection, and then you can still continue to just focus on the razor and what it is that's going in there. I don't know that that's going to happen, but the idea is, you know, using what we can to define the important characteristics of that razor handle, like making sure it doesn't weigh a ton and you know, and it's not pointy, whereas what color it is maybe just doesn't matter because it still holds a razor and it's still fine. So being able to determine which ones are functionally important and which ones are not functionally important, and then incorporating that in the FDA's view of the world, as well as who can make those vectors for delivery. I don't know, maybe I've taken it too far, Peter. I'm sorry, but I do like the, you know, the idea
No,
of thinking of it
it's
that
great.
way.
Yeah, you're just fine. All fine. No shaving next.
There's a question from Dr. Pan around, which I want to make sure we are very clear on this. The sweet range of 100 to 1,000 doses of GT production, wondering the percentage of diseases that were not prioritized because of this restriction. I just wanted to start before I let Peter address that, is that I mean, our main focus here absolutely are those in the much lower range, the ultra rare, the bespoke, where there's no commercial value. But by choosing that window as opposed to another, certainly think there's learnings that can come out of here that certainly will add to, you know, larger in the, I guess, the rare disease versus the ultra rare disease capacity. But our focus is going to be on you know, the much lower amounts.
No, I can say that at least from several companies have shared with me their assessment of what fits into the 100 to 1,000 range. And although, in other words, that commercially viable range, and it's not as large as you might think it might be, because many of the diseases are just below that, at least in one country. Obviously, if you aggregate globally, maybe you'll get above that. So we actually did think about this a lot. And so we think that, you know, one of our kind of teasers of a disease might be to try to address disease where it's perhaps half at commercial viability, like 50 patients per year, or would need 50 treatments per year. Because the idea here is, if you made that much more efficient, could you bring it over the finish line? In other words, if half as many doses, but twice the efficiency could be the same thing. So just to bring up that, you know, I think this is a potential way of moving forward.
I think a good question that really resonates well with what AMP has been is, can we elaborate on that the AAV biology portion? There's certainly a lot of existing NIH funding opportunities, even FDA funding opportunities around advancing AAV biology, and the need to require that through a public-private partnership to succeed, and, you know, versus simply just being an NIH effort or a government effort going forward. What is the, you know, the core benefits there for public-private partnership? Joe, I might ask you to make that comment too, but certainly in the AMP world, that has been a very positive mechanism to expand, do more, raise all boats, and I certainly think the private side of things also greatly benefits and wants to see these advances, and it enables them to contribute in a greater way and really accelerate and kind of raise the bar of some of the research that's being done. But I'll ask others to comment.
So I'll just add to that before PJ jumps in, because I saw them starting to take a breath. One of the things that we have found in other AMPs, as well as other partnerships that we do at the FNIH, is not so much that it changes the rigor, it changes the focus. And I think our private partners, including FDA, but who's not private, but you know, really have an intense focus on those advances that will be more generally applicable to the entire hire, you know, what they need in manufacturing or they need in whatever, which will then you know, be generally applicable to the field. And I think that focus consistently generates very actionable and executable results. And so you know, to be semi-glib, you know, much of the AAV biology could go out as being a standard NIH grant. I think what the private partners do is make sure that that has a defined applicability to the production, in this case, of a vector that can be used for people. There's a lot of things that aren't really too bright and shiny sometimes about some of that, but that's the kind of stuff that really gets you over the finish line for generating something that can go into people. And so I think that's what I would say is the partnership provides a level of focus that really a standard RFP doesn't generally.
Yeah, I guess I'd just say too that theoretically some of this could be funded by NIH, but all of the NIH institutes and centers have their own funding priorities, and you know, is something this specific going to be one of those? It's not truly basic research. I mean, it's basic AAV biology about how to make better AAV vectors. So it could be done. I'm just not sure it would be done. And I think this is a wonderful opportunity to see this, I think, really critical data be generated, you know, through this partnership.
Peter, anything else from you?
No, I just, yeah, you're good.
Very good. And then before we move on, one last question gets a little bit of detail, may not be ready to answer, but I appreciate the questions that are coming, because a lot of your feedback and your comments and the questions that you guys are bringing up is going to help us make sure that we answer these things, make sure we're prepared for when we reach out a little more formally. To make sure we clarify in a very detailed and executable project plan to make sure we answer some of these questions. But there was a question Scott Galinsky sent: what is the expected cost of each of the BTGC's therapeutics produced? Right, I think those are going to be difficult based on what we decide and how they're manufactured. But if we can address how we'll make those decisions and what will be involved there. Peter?
PJ.
Yeah, I don't really know how to answer that, because I think it obviously depends on whether you're making, you know, how much vector you're making. Is it one for, you know, a disease where you have to treat the muscles and essentially treat the whole body, or just for the CNS, or just for the eye? So I don't know how I could answer that in general.
Understood. I think a lot still to be determined before we can figure that out. Sam, just popped up a question about having us elaborate a little more on the governance model and the decision-making process, that we can ensure the speed of this, the accelerated development of this. Joe, I'll defer to you, but I'm happy to weigh in also.
Yeah, so the governance model is that of our current AMPs, and it is really, while we have an executive committee that we go to help smooth bumps in the road, or as in Peter's slide, knock down some barriers, we really rely on our steering committee, which is the folks made up by, you know, the supporters. Those who support the partners who support the project, and they have a lot of the responsibility for okaying, you know, RFPs, particularly those that are being funded through FNIH, and defining what gets done and what could be done. And so in general, what we try to do is make sure that the governance stays out of the way, but is supportive of making things move in the defined direction that we need to go. It also is very, because it's generally a small 20 or 30 people, we can make decisions relatively quickly about whether something actually has reached a milestone or not, and whether we move forward, or whether it should be, it's gotten to a point where it no longer appears that it will fulfill the requisite endpoints, and so we can shift that to yet a different, shift that resource that could have been used or would have been used to continue onto another program or project. So a project within this one, right? So I think the idea of the steering committee with some working groups under it that report to the steering committee is really that's the project team that is making the decisions about what gets done in the overall project.
And that provided a good transition to our next section, that we really want to go through a couple of these opportunities and actually what that partnering and those responsibilities may well be. So at least at the highest level, I think we'll touch on this. Joe, am I defaulting to you here or taking the
Sure.
first couple?
Sure, I can take the first couple and then we can switch back. So if you next slide, we're going to talk a little bit about now, just very highlight, there's more words here than I will actually certainly read, but the point of these are that within the project, we have already begun to think and will continue to think about how to communicate these results to advance the education of and engagement strategies in gene therapy across the field. And so we will use communications to help fund and develop. We will have some, approaches to external stakeholders as we see. We'll look for internal stakeholders that can continue to communicate and to AMP about what AMP GT is doing. We include our partners' communications groups as well, so that we make sure that what is being provided is clear. We also will support the outreach to any working groups that are necessary. And then finally, again, if there's a need for patient recruitment. These are just a few of the kinds of things we do. It's just to give you an idea of what kind of things we are at our disposal, but not necessarily that we have to do any of these things. We do the things that make sense, and we do the things that make sense to the project team and their communications groups as well. The next slide is development, which in terms of how we look at this, and how we obtain the resources in order to run the project. In the past, and we will continue in the future to incorporate our message in as many places as we can. This is a slide that shows lots of web pages, because obviously nowadays that's the way to go. And in fact, the top one, the top left web page there is for PAVE GT, which if you look at this, it's kind of a version of what we're talking about, only in slightly more limited, which is a great web page. So we will expect to have the kind of web presence necessary to make sure that people know what we're doing, and that people learn as we do, as we move forward. And the idea is that we will complement and link resources as many and as often as possible. And finally, actually, the next slide addresses a question, actually, a number of questions that came through in the that we didn't actually, so sorry about that, but we knew that we were going to talk about it. in all of our projects, we are acutely aware of what's going on in the outside of the AMP structure. And particular IMI has had something going on in gene therapy for as long as we've been working on this. And we specifically avoided duplicating or doing anything that we thought would directly overlap with any of the IMI approaches. That's not always 100% possible, but we try as best we can to complement one another. It's even easier in this case because IMI has a very active and open view, or active and open relationship with the Heaver group that I pointed out at the beginning is part of which is the heads of R&D that has helped this get started. And so it's brought up there frequently. What's going on in AMP, what's going on in IMI, let's make sure that we're doing things that are complementary and not duplicating efforts. I think the next slide goes to what we are thinking currently, and it's described again what we were talking about in the budget slide, and just to be absolutely clear, again, 102 million over 5 years. That's the current budget, but it's a estimate. We expect NIH to provide half and the private sector to be half. If we had 10 large pharma partners, we're talking about $1 million a year over 5 years, for $5 million a year for 5 years. And we can have some discussion around tiered levels of investment, particularly for other nonprofits and things like that. We've done that in the past, but we do realize that the major partners and other AMPs have been very strongly firm about the idea that we should have equity, and if you're in, you're in as a full partner. But again, those are discussions we will have as we move forward. Full voting on the EC would be a standard R&D stakeholder, and then there are other opportunities that we would address for other organizations. And then I guess the next slide. We are proposing that we will also have not-for-profit philanthropists and advocacy organizations as part of the partnership. This support, obviously, for not-for-profits and advocacy organizations, et cetera, is not the same. It's not going to be the same as the Pfizer's and Merck's and others. So that's and that will follow probably follow something like this kind of tiered support, and then. And you can read through this, but again, some of this will be very firm, as we described, and others we will be able to have a little bit of opportunities for negotiation. Steve, do you want to take the next couple of slides, because I've talked for a while?
I can. And thank you. And I can clarify, update on a couple other points too. As what's always been part of AMP, and we outlined earlier that this is a kind of a 50-50, not only in its funding support, but also in its governance. And the way that this works is 50% of the vote is really on the government side, and that's inclusive of the not-for-profit partners, and then 50% is on the industry partners. So that's the way the vote works in the governing model for how we do this. Certainly in many aspects in sub-team or as in ex officio members of the steering committee, we may have key academic investigators or some of the leads that are, the awardees that might be from the academic sector. They're certainly involved, but obviously to avoid some potential conflicts, you know, they're not going to be direct voting members on how we allocate that funding or all that for them, but their expertise and their weigh in an ex officio capacity certainly can be highlighted and involved in the program. Next slide. And then it's always important to understand what this is, you know, what's this going to come to bear on to be one of these representatives. What we've typically done is the steering committee meets in a monthly capacity, getting slide updates and making these decisions and monitoring and guiding the progress through the program. We often have 1 to 2 face-to-face meetings per year. When it's a non-COVID year, we often have these at national conferences and can get together on an annual basis. I think we spoke a little bit about RDAT and the IMI effort, and I think you're going to see a concerted effort where there's other programs to invite or co-invite those to some of these face-to-face meetings, or perhaps have workshops and offshoot symposia where we're able to align and share some results and share findings and co-learnings together as we go forward. And then obviously we talked about the clinical development side of this. We talked about some of the other opportunities involved in interacting with the regulatory element, the manufacturing element of this, some of the communication efforts. There's going to be some sub-teams, some groups where additional representatives from your organization certainly could participate and then weigh up the information to the steering committee if there's any type of decisional vote. But those opportunities for greater engagement are going to be there, and it's going to be driven by the need, the science of when we need to go to a lesser level. And I don't need to jump back to the slides, but I did want to also say that one of there's been a question, and we're going to move into that Q&A period if there's additional questions on partnering here, is that Joe talked about tiering, and we understand that at the what we'll call the larger pharma, manufacturing pharma type of level commitment that we're going to do to support the project, that's certainly going to have a particular threshold, but we well recognize that there are other tiered levels of smaller gene therapy developers that could get engaged in this, and we will work with some flexibility of what that tiered structure looks like and what the equal access and equal sharing and role in voting will be, and we'll outline that certainly in the plan. And we also saw, some of the non-for-profit, some of the philanthropic and advocacy groups, we want those to be key components of some of these communication efforts and a strategy, developing that strategy, helping us with the content, reaching out to the communities, as far as reaching out even in patient recruitment and identifying those. That's going to be a critical element. So we want to make sure not only do they have the opportunity to weigh in with their expertise, but also be involved in that decision making and be involved in the steering committee updates. Those that might not meet that threshold still are going to have the opportunity for progress updates, still have the opportunity. We have what we call an AMP extended executive committee where we do some biannual updates on all the AMP programs, provide progress reports, provide those things. So we develop that governance, we develop the sharing and access in line with the partnering opportunities. And I think you'll see that spelled out in a detailed plan a lot better. But we don't want to close doors from expertise. We don't want to close doors from people that could really help us in the different segments of the program. So with those types of comments, we did have another short window for Q&A. We really had a good opportunity to address a lot of the questions. I know there may be others that come up, and what I think we will do is capture those from the chat. We will make sure that the FNIH works with the presenters and the other leaders of the program to get back to you individually where we might have missed one of the questions and follow up. And I'm going to address here in a second some of the final steps in contacting us if you do have additional questions. So my suggestion is we move forward, and that'll answer some of the other questions that are popping up. Next slide, please. And the next slide, please. So I just wanted to reiterate where we are, as Joe outlined in the beginning. Right, we are at a point where we've shared with you this concept document. A lot of that information's on the red site that we sent out. We're going to share these slides, but we're at the point of taking what we've got from a lot of those working groups and putting that into a detailed research plan to move forward. I'd move to the next slide. So not only as an action item out of this webinar, as I spoke to, we're going to take those questions and follow up and make sure that we get anything else that you guys have as questions and comments and provide those answers to you. But there are some key things we're still doing, working on that pilot manufacturing process, how exactly we're going to engage with the BGTC and what that will look like. And same thing with the interactions with FDA on the clinical development side of this. The same thing with refining the disease selection criteria, working very strongly with the bioethics group at NIH to define that process that we would carry that forward. And then ultimately that will lead into final decisions of what goes into the pilot. And then thinking even forward beyond the program at a later time period. And also, you know, I talked about the communications efforts, and Joe touched on that, and continue the development of that strategic plan and engaging folks that have expertise and know the field and know the advocacy organizations and can tap into that to help us in defining that project plan. That really is what needs to happen, and we're going to do that through the end of Q3 of this year. In the beginning of Q4, we hope to be able to share that project plan with potential supporters of the program, and that will beget us reaching out for letters of agreement in the fourth quarter of this year, asking for support and funding for the program. So that's when you're going to see FNIH reach out directly to your organizations to meet these levels of support. And again, we'll have that finalized project plan. A tall order, but certainly we have our pedal to the metal to get that pen to paper and get that done and completed and out to all of you. Before I step to the last couple slides, Joe, I didn't know if you wanted to add anything more to that, or the same thing for Peter or PJ on finalizing that plan and meeting those timelines.
So I think this is, you know, as I said before, it's an ambitious project. It's gone a long way. It's actually the concept is quite. It has a lot of good framework, but it needs to be put into practice. And so, you know, some of the questions that have been coming through is just like, when will you actually start putting together the partnership? This webinar is the start of putting together the final partnership, and so we are now in that space, and look forward to, you know, bringing in as many people who will be interested in making this happen as possible. It's a unique program from an AMP point of view, which is why we started with variations on a theme, because of the absolute need for the FDA to be involved, the certainty of the connections with advocacy groups and other not-for-profits, and the potential for bringing in philanthropic additions, including, as Peter reminds me, venture philanthropy that would allow us to you know, get something put down on the ground and get this out. So I think this provides a unique space for that. That gives us a little bit something new to think about at FNIH, but from a project point of view, it just means it has that much more, that many more stakeholders that can produce a better product in the end. A good manual, a good process.
Peter, PJ, anything there?
This is Peter. So I just wanted to thank everyone who's joined. I think this, to me, it is one of those things where there is tremendous opportunity here. One of the things that I find most exciting about being at FDA is that we get to see the entire spectrum of what's out there in a given area, and seeing that, and then seeing what we're trying to do here. This really does, I think, fill a very, very sore need in this area that could make a very big difference for a group of people right now, while also really plowing the road for where we're going to be going as we have more and more very niche gene therapies and where we might go with CRISPR-Cas9 constructs, et cetera, down the line. So I think this can help us get the regulatory paradigm straight. It can help us get the non-clinical work straight, the clinical work straight, and actually the manufacturing that needs to support this all, which really is one of the most limited pieces of this right now, or limiting pieces of this. So I'm just so grateful for your interest, and we'll really look forward to working together, and I hope this moves forward. Thanks.
Yeah, I'd just like to echo Peter's comments. Thank everybody for participating. I think the questions are really valuable and gave us some additional food for thought. So thank you all. Just
move to the last slide. So again, just so you have the contact information for us at the FNIH, we have multiple parts of our internal groups that are involved in this on the research partnership, Joe and I, the comms side, and on our development and fundraising efforts by senior leadership in this program. So a lot of dedicated and senior level involvement at FNIH to make this successful and to be engaged. And you'll hear from us, and we hope we hear from you. So if there's follow-up questions, follow-up comments, opportunities to engage, you're going to hear from us, from these folks, and these are the right people to reach out to from your side. So we look to that exchange going forward. And then the last slide. We thank you for your patience and listening in, taking up 2 hours of your time to hear this. So very much thanks for your attention, your engagement, and we look forward to hopefully partnering with you with this important, impactful program. Thank you today, and have a great day. Stay safe, stay healthy.
Yeah, thank you all for all your questions. They will be very helpful in continuing to move this forward. And I hope we will be talking to many of you very soon.
Thank you.
Thank you again.
so much.
Have a good day.
Bye. Thank you very much.
Bye-bye, everyone.